This protocol aims to isolate the epithelial parts of the mammary gland free of associated mammary stromal cells. Begin with a crude dissection of the surgical discard material to enrich for epithelial structures. Using enzymatic digestion, separate the epithelial organoids from the stromal material outside the basement membrane. Then, process the digested organoids through fixed pore size cell strainers to obtain enriched fractions. Seed the organoids for immediate culture or freeze stocks for future use. Ultimately, the epithelial organoids are used to generate long-term cultures of normal human mammary epithelial cells.
I first developed this method in the late 1970s in order to advance the field of human epithelial cell biology and carcinogenesis. Abundant surgical discard material from normal human mammary tissues was available. What was needed was a method to process this bulk heterogeneous material to make it suitable for pure epithelial cell culture, and media to support long-term growth. Visual demonstration of this method is helpful, particularly for noting how to do the crude dissection, what properly digested organoids look like, filtering the organoids, and plating the organoids for culture. The advantage of this technique is that it provides clean epithelial fractions free of stroma, and the epithelial cells remain as intact organoids, which we have found gives the best initial growth and maximizes the number of cells that can be obtained. Two scientists from the Berkeley National Laboratory, James Garbe